Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on dark reaction. So it is called dark reaction. Why? Because it is light independent reaction. That's why it is called as dark reaction. So it was discovered by Melvin Kelvin. The scientist Melvin Kelvin identified this reaction. He discovered this reaction. That's why it is called as Kelvin reaction. Okay. So the first stable compound, that is 3-PZA, which is a 3-carbon compound. That's why it is also called as C3 cycle. So the ATP and NADPH, these are nothing but assimilatory power. They are generated in the light reaction, are going to utilize in this dark reaction for the reduction of CO2. CO2 here reduced into a carbohydrate. Okay. So it is a primary pathway in so many photosynthetic prokaryotes as well as eukaryotes. Okay. So the site of dark reaction is stroma of chloroplast. Due to the pentose phosphate involvement, this cycle is also called as reductive pentose phosphate pathway. So, RPP pathway. Due to the reductive nature of this cycle, so here the reductive reactions are going to happen. That's why this cycle is also called as photosynthetic carbon reduction. Nothing but PCR cycle. So these are the alternative names for C3 cycle. So let's draw the C3 cycle. Then I will explain step by step reactions. Okay. Okay students, this is the dark reaction. I will explain each step. Okay. So 3CO2 in the first step, that is nothing but carboxylation phase. So this first step is related to carboxylation. Here 3CO2 is accepted by RUBP. 3CO2 is accepted by 3 RUBP molecules, ribulose bisphosphate molecules. So in presence of an enzyme Rubisco. Okay. So it converts into an unstable 6-carbon compound. So it breaks quickly into 3-phosphoglyceric acid, 3-PZA, which is a 3-carbon compound. 6 molecules of 3-PZA are synthesized in the first step. So that step is nothing but carboxylation. For our easy understanding, so the dark reaction is divided into carboxylation, reduction and regeneration. So let's say this is the first step. Okay. So in the first step, it is 3-PZA is generated. Okay. So in the second step, the 3-PZA is going to phosphorylation. The second step is nothing but phosphorylation step. In presence of an enzyme kinase, 3-PZA is going to convert into 1,3-bisphosphoglyceric acid. 3-phosphoglyceric acid is converts into 1,3-bisphosphoglyceric acid. So in this step, 6 ATPs are utilized. Okay, 6 ATPs are converted into 6 ADPs. Okay, so 6 phosphate groups are added to the PZA molecule, then it converts into 1,3-bisphosphoglyceric acid. Okay, 6 molecules of 1,3-bisphosphoglyceric acid is generated. Let's see the third step of dark reaction. Here 1,3-bisphosphoglyceric acid is converts into G3P. Okay, glyceraldehyde 3-phosphate with the help of 6 molecules of NADPH2 is nothing but reducing power generated in the light reaction. Here 6 molecules of NADPH2 are utilized. Okay, so 6 molecules of glyceraldehyde 3-phosphate are generated. Here, among 6 molecules, only one molecule of G3P is going to cytosol for the preparation of glucose. So G3P is a 3-carbon compound, whereas glucose is a 6-carbon compound. For this reason, this cycle has to occur 2 times for the 6-carbon compound preparation. Okay, if RUBP accepts 3-carbon compound, so the cycle has to occur 2 times for the sending of 3-carbon compound. Okay, for this reason, the cycle has to occur 2 times. Okay, so remaining 5 molecules of G3P, they undergoes regeneration reaction. So second and third reactions, they are related to reduction reaction. Okay, first one is carboxylation. Second and third reactions are nothing but reduction reactions. Here the 3PGA is going to reduce into G3P molecules. Among 6 G3P molecules, one is going to cytosol for the preparation of 1 glucose. So remaining 5 molecules are going to regeneration reaction. They are going to regeneration reaction for the preparation of RUBP. For the regeneration of RUBP. Okay, so 5 molecules of G3P, they converts into ribulose 5-phosphate first. So which is a 5-carbon compound. Now the ribulose 5-phosphate again converts into RUBP with the utilization of 3 ATP molecules. Okay, so ribulose 5-phosphate converts into RUBP with the utilization of 3 ATP molecules. So the dark reaction involves 3 main steps. They are nothing but carboxylation. First step, RUBP accepting carbon dioxide, that is nothing but carboxylation. There it converts into 3PGA, 3-phosphoglyceric acid. Second step involves the reduction reaction. There the 3PGA is going to convert into G3P molecules, glyceraldehyde 3-phosphate molecules in 2 steps. Okay, this is the first step. There ATP is utilized. In the second step, the NADPH2 are utilized. Okay, so 5 molecules of G3P molecules, they are going to regeneration cycle. They has to regenerate RUBP for the continuity of C3 cycle. It should occur continuously for the reduction of sugars. That's why, so 5 molecules of G3P molecules, they are going to regeneration reaction. They converts into RUBP molecules again. Okay, so let's see the energetics of the dark reaction. Energetics nothing but how many energy molecules are utilized in the dark reaction. Let's see. Okay, so in the phosphorylation step, 6 ATP are utilized. Okay, 6 ATP are utilized. In the 6th step, in the regeneration reaction also 3 ATPs are utilized. Okay, so in the 3rd step, 6 NADPH2 are utilized. So total 9 ATPs are utilized and 6 NADPH2s are utilized. We have taken 3 RUBP and 3 CO2 molecules. So the cycle has to occur 2 times. Okay, so 9 into 2, that is equal to 18 ATPs are required for the synthesis of 1 glucose molecule. Okay, so 6 into 2, that is equal to 12 ATPs are required for the synthesis of 1 glucose molecule. Okay, for every turn, if you take 3 carbon dioxide, for every turn, one 3 carbon compound, that is G3P, is separated from the cycle. It is going to the site salt for the preparation of 1 glucose. That's why the cycle has to occur 2 times for 3 molecules of CO2. But for in real, 6 Kelvin cycles has to occur. Each time one RUBP accepts 1 CO2. In that case, the cycle has to occur 6 times. Okay, students, this is about dark reaction. Thank you.